Thanks, Asia. So today we have the pleasure to have Dr. Agustin Ibanez. He's a neuroscientist interested in global approaches to addressing diversity and disparity in brain health. He's a full professor and director of the Latin American Brain Health Institute at the Universidad of Adolfo Ibanez. Also, Agustin is a professor in global brain health, and he directs the international program in global brain health research at the Global Brain Health Institute, Trinity College Dublin. He has a team, and he's a team leader of the Predictive Brain Health Modeling Group at Trinity College Dublin, a senior Atlantic fellow at the Global Brain Health Institute, and a research full associate professor at the GBHI Trinity College Dublin. So today, Dr. Ibanez will talk about the exposome of brain aging, and I'll turn back to you, Augustin. Thanks really for presenting the talk.
Thank you so much, Fadi, and all of you attending today. As Fadi mentioned, I will talk about the exposome. Those are my disclosures, and before jumping into the exposome, I just want to make the case for a larger discussion about how we may want to face, probably in the future, the interaction between environment, body, and the brain, that I call a brain health synercom. I'm trying to bring this idea of neurodynamics, and then exploring the connections between body and environment through exposome and whole body health research. And I will try to then assess the overall relevance for future research and also for some policy oriented guidelines. So the first thing is that I invented this term that called brain health synercom to try to make the case, to try to start to connecting the dots between body, environment, and brain. That sounds extremely obvious and simple, but it's much more complex than appears. The fact is that we have genome, that is the totality of genomics influence. We have the metabolome, that is the total metabolic influences on health. We have the exposome, right, that they're the total influence of the environment on health. But when we define this, we tend to think that these things are happening in isolation. What they are really, interconnected in real life. So the environmental factors interact with the metabolome, with the exposome, with the genome, with many other physiopathological pathways. Unfortunately, we have learned to divide and conquer, and we have ignored these connections. We are starting to do connections, let's say, between genetics, epigenetics, and environment, but we we really don't understand how is the massive interactions between these factors and how to model that. Most of our models are not close to trying to address these things that are kind of circular interactions, spatiotemporal dynamics, non-linear interactions with different, completely different landscapes with subtle combination of environmental versus whole body and brain health. So even when we don't have such a model of the brain health synercom, I think that we need to start to connecting the dots, and I will try to make the case for assessing some strategies for doing that. The first thing is trying to putting back the brain into the body, to say it simple. I have been always trying to understand the cognition in the wild. When I was an early neuroscientist, I was very interested in the kind of cognition that we study in the labs. It's a very weird or atypical way, stimuli, isolated stimuli with a lot of repetitions without backgrounds. So we have ignored the cognition in the wild until, I don't know, the last 10 to 20 years that we started to incorporate. But the same kind of moving to, reincorporating body and environment into the brain is also relevant for brain health. And this is that we have been trying to push with some theoretical accounts of that. if you think in the One Health approach, is the perfect model to try to understand how the environmental factors impact the body and how the body impact the brain and how the interactions between these layers are essential. So when we think in physical exposome like pollution or heavy metals to social exposome like education or socioeconomic status, these factors are really creating a kind of biological embedding in terms of epigenetics, allostatic overload, and inflammatory process, and many other processes. And we need to start to understand that. This is important not only for the brain, as we were talking, but also for cardiovascular, cardiorespiratory, visceral, and musculoskeletal structures, in which the environmental demands across the lifespans are always a dynamic system that we need to start to understand. Just to understand the process of, let's say, an organismic interaction with the environment, the concept of allostasis is a very simple one that suggests that, our brain is constantly trying to update the information about the context and trying to anticipate what's going to happen in both terms of cognitive aspect, emotional aspect, but also in terms of responses to pathogens or to different health threats. So let's say that our visual system captures some, risky situation, or brain, increase the breathing rate, the heart rate, the muscle tone, and also this allows us to, provide an adequate response to an environmental demand. When we have different stressors, like, when you miss a train or you lose your keys, this allostasis increase in your body. And this is a normal oscillatory pattern in which we tend to, respond to environmental demands and then come back to a kind of baseline. But then when the kind of stress accumulates, we have what we call allostatic overload, or like, starting of atypical responses in the brain and in the body that increase inflammation, for example, increase the risk of diabetes, of higher body mass index, etc., 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 as well as mental health issues. This is important because we tend to think that environmental factors, being physical or social, are like a kind of additional impact in the brain, while in fact they create from, body health, like cardiometabolic health, to lifestyle factors like exercise or sleep deprivation, to social factors and to physical factors. They are all stressors that can have a real biological embedding. Just to give you a very small example how macro factors can impact micro level of the biology is that, the allostatic load model in Alzheimer's disease, in which, stress, environmental stress, and internal stress can really impact, the very small microarchitecture, like inducing reactive astrocyte or impaired insulin signaling or activated microglia. This process at the same time then accelerates a neurodegenerative process. We have been studying the allostatic interoception, like the brain's ability to sensing, monitoring, and using body signals to respond to environmental demand, especially in conditions like frontotemporal dementia, in which we have shown that the atypical body responses are an important hallmark of autonomic and interoceptive deficit in this condition, associated with many clinical, cognitive, and functional outcomes in frontotemporal dementia. Also, when we look at, meta-analysis of more than 5,000 participants across many different studies in neurodegeneration, when you see deficit in interoceptive signal, you tend to see deficits in emotion and social cognition, suggesting once again that the interoception is really critical for cognitive as well as clinical aspect of the disease. With George Northoff, we have started to develop some mathematical models to starting to identify interoceptive signals and linking them to other clinical and cognitive aspects of brain health and disease. For instance, in this paper, we use this We measured the interoceptive cardiac symptoms, and then we shown that they were associated with clinical, cognitive, and neuroimaging correlates on frontotemporal dementia. And, also studying neuropsychiatric symptoms, that many of them are expressed through the body, right? We shown that there is a accelerated aging in the brain associated with neuropsychiatric symptoms, especially in the allostatic interoceptive network. There are many other approaches that are pointing to this. So Tian and colleagues like Jung Hao Wen and many others working with multi-aging clocks in the body have shown the relevance of studying the whole body. Like, with 3 of the, let's say, cardiovascular, hepatic, or musculoskeletal organ clocks, you can better predict neuropsychiatric symptoms than just looking at the brain. And we have shown also that the peripheral, like the cardiovascular risk factors from the body, have a strong association with the structure and function of the allostatic interoceptive network in different dementias, but of course, especially in frontotemporal dementia. This is important because the body is the window from which the environment have a hallmark in health. When we think in diversity, like age, education, genetics, ethnicity, etc., and very important underlying factor is disparity, like socioeconomic adversity, early adversities, low education, poor cardiometabolic health. All these factors impact through the body into the brain. And for me, it's very important to understand how the environment can leave different pathways that can be assessed at whole body, but then in the brain, right? So let's start to summarize some of the evidence. For social exposome, the combination of different environmental factors that are mediated by others that impacts the overall health, let's say education or social interaction or loneliness or socioeconomic aspect, all of these factors have a huge impact in the health. We have been done multiple research. For instance, we compared the brain, the structure and function of the brain in aging in US versus Latinos population, and we have very large differences. But an important amount of this difference were just explained by educational disparities. So just different in years of education have a hallmark on the structure and function of the brain in aging and dementia. And when we look at the not just of year of education, but also qualitative education, those impact were 7 times larger than just education, suggesting that the quality of education is a critical, probably through cognitive reserve aspect that impacts the structure and function of the brain. Also, socioeconomic inequalities, those, the higher, extremes level of poverty or richness within a city or a country are associated with multiple metrics of brain health, including, typical EEG measures of connectivity, oscillations, periodic and aperiodic measures, or entropy measures, for instance. The same is happening with fMRI, and we have shown that the structural inequalities linked to the structural and functional connectivity of the brain in aging and dementia. And this is more accentuated in underserved populations like Latin Americans one. But then trying to bring a more complex idea of the social exposome, we developed an assessment with more than 300 questions about education. For example, education of the parent, the quality of education, private or public, food insecurity, financial status, assets, access to healthcare, subjective socioeconomic status, childhood experience, and others across the lifespans. And what we have shown is that these social exposomes have strong associations with mental health, with clinical and cognitive aspects in healthy aging, but also in dementia, and also association with the volume and functional connectivity of the brain. Also, we have shown that these social exposome, in terms of social vulnerability, have a huge impact across the deep phenotyping of clinical and cognitive process, including, global cognition, memory, language, attention, relation, executive function, visual processing, theory of mind, emotion recognition, functional abilities, depression, anxiety, and neuropsychiatric symptoms, for example. And this is consistent across different countries in Latin America that we have been assessed. So this brings us coming close to the idea that the exposome is not one factor, it's not a risk factor. It's an interacting set of multiple factors that have, different nonlinear interactions. This is the idea that I want to come later on. So the first thing that we did, we looked at the brain aging using convolutional neural networks to estimate the accelerated or delayed aging across 20 countries. And that we found is that the people that lives in the south, in the global south, tend to have more accelerated brain aging. Females tend to have more accelerated brain aging also. Uh, um, People with dementia also have more accelerated aging, but the combination of these three, so if a person have dementia, comes from the global south, and is a female, will have the largest accelerated aging. We started to explore the impact of the environment in this case by looking with association of socioeconomic status and pollution, and we have that both components, physical and social exposome, were strong modulators of this accelerated brain aging. Later on, we developed a biobehavioral metrics of aging in which we compared protective factors like cognition, functional ability, well-being, education, or physical activity, as well as risk factors like heart disease, hypertension, sensory impairments, and lifestyles to identify the people that have older than expected or younger than expected age. What we found is that the people in Africa, followed by Latin America, have the largest accelerated aging, followed by Asia, and then European countries, with the northern European countries having the most healthy aging. At that time, we started to explore association between different metrics like pollution, or socioeconomic status, or even sociopolitical instability, and we found that they have similar impacts in associations with accelerated aging. Also, we use this biobehavioral age gap, the same that we're using in this study, to show that the people that looks older tend to die earlier and to convert to dementia at a more rapid pace than the people that have less accelerated aging. We also shown across 33 countries that the people with accelerated aging tends to go more frequently to the hospitals, and this, of course, was modulated by socioeconomic status and other factors. More recently, we started to combine the different metrics of exposome. So in this time, we compared 73 metrics, including air pollution, temperature, climate disaster, different soil, water, air quality, as well as social factors like, energy demand, unemployment rate, democracy indicators, migration, etc. And we look at the individual contribution or contributor of these 73 exposome, as well as to the nonlinear interactions using GAN models, generative additive models, and that we found that the different individual physical and social factors were associated with accelerated brain aging. But when we have the combination of factors, we have a order of magnitude of 15 folds larger association with accelerated aging. This is very intuitive. That suggests that poverty, for example, is the kind of syndemic effect, the kind of synergetic effect in which multiple factors induce a much larger accelerated brain aging. This was present. This effect, of the environment was comparable to the accelerated aging observed in patients with dementia, and we started to try to understand what is driving this accelerated brain aging. For that, we use biophysical semi-empirical models in which you can use the empirical data and feed this data with nonlinear equations models to try to understand the underlying mechanisms and basic mechanisms associated with unhealthy aging, like structural excitatory inhibitory balance of the networks and other mechanisms were associated with these brain clocks. In this paper that is going to be published, I hope soon, we created the first biophysical brain twins in 7,000 participants. We created more than 3 million of simulations to create for each subject a synthetic brain twin of the brain dynamics as well as the basic mechanisms in terms of entropy, excitatory, inhibitory balance, and other metrics. And with that, we've shown that both the demographics or phenotypes, including sex, age, or diagnosis, healthy aging versus dementia, as well as expotypes like exposome-related aspects, including socioeconomic, migration, demography, air pollution, green space, temperature, precipitations, soil and water quality, climate disaster, or others, were associated not only with, brain dynamics, but the twins, the biophysical properties of these dynamics that were modeled across individual level. This is also an opportunity to understand not only environmental factors, but also genetics, and I hope in the future interactions between genetics and environment. So, for example, we use Down syndrome, that is a hallmark model, I would say, of accelerated aging to estimate brain clocks and to develop biophysical model of that. What we found is that the Down syndrome have more than 25 years in average of accelerated brain aging that were associated with, hypoactivity, reduced excitability in the patients, except for those at very early stage that are showing hyperexcitability. This is a pattern that was shown in multiple animal studies. And then we look at the spatial transcriptomics and found that they were a cascade of genetics of neurodegeneration associated with the excitation of the brain areas that were most associated with accelerated aging. Then we develop perturbation analysis to explore therapeutics, but I will skip that part. So the overall idea is that we really need to, when we study the brain, we need to look outside the brain, not just inside the brain. And we need to look at the whole body health through interoceptive cardiometabolic pathways, as well as to the impact of environment and those interactions. So this is I'm so proud to be the founder and co-director of the Red Light 2 initiative, a large NIH founded in which we will explore the genetics and exposomic interactions in aging and dementia across 13 centers in the Americas, including US and Latin America. But also we have been pushing the agenda, trying to say, if we really want to understand the exposome, we need to move beyond understanding individual risk. We need to understand the nonlinear, syndemic, synergetic, emerging interactions between factors. For that, we need, a kind of different methods that traditionally have been used. And we need to map in the exposome with much more granularity. Happy to discuss more about that. Whole brain generative models are important because not only because they provide more, let's say, organismic or whole level dynamics of the brains that allows to understand, principles of micro dynamics into global dynamics. So you can develop manifolds that allows to understand global properties of the brain without looking at the millions of small individual interactions. And you can, combine priors of atrophy, structural connectivity, metabolism, neurogenetics, or cytoarchitecture to develop, more robust models. But at the same time, you can use order parameters to understand the impact of body health, like microbiome or inflammation, or environmental factors like socioeconomic disparities or pollutants. So I think that whole body generative whole brain models are important for the future of this agenda. And also, I think that the allostatic interoception is a concept that can really help us to understand how different environmental factors, including physical toxins like pollution, but also social toxins like stress or, violence in the environment, can impact different inflammatory, immune, metabolic, and microbiome pathways across the lifespan, right? And having a cumulative burden that can be understood in that regard and help us to understand also, transdiagnostic effect that sometimes we observe between anxiety, depression, Alzheimer's, or other conditions like frontotemporal dementia. We need to really better understand the interoceptive signal, how social factors impact the cardiac and body signals in the brain. That's our example of that. I won't go into the detail, but happy to discuss later. But also to understanding that not because we put the focus in environment, that means that genetics or biological interactions doesn't matter. On the contrary, I will say the vulnerability to environmental factors can be really amplified or reduced by genetic influences. And this is very important because we can have, examples of resilience and also higher vulnerability across different populations, and understanding the interactions between genes in terms of not only, of mutations or founder effects, but also more diffuse effects like epigenetics and polygenic risk score are really important to understand these lifespan interactions between environment and biology. I think that this is beyond dementia, and it should be an important approach for overall brain health, both mental health and neurology. And this is also important to understanding, not only for understanding disease, but also to understand health and why there are people that come from extremely poor environments or extremely, poor cardiometabolic conditions and still develop resilient brain health. I think that resilience needs to be understood not only as a genetic or brain, but also cognitive, cultural, environment, and social aspect. And the interaction between these levels are really important if we want to develop a personalized model to understanding of resilience. We have proposed aging clocks because you can leverage the impact of expotype, like all the different physical environment, as well as phenotypes, like in terms of psychological lifestyle, etc., how they impact different aging clocks. There is a long discussion about what is aging, and there is no agreement between the experts, right? And I think that there is no agreement because they are not a single model or a single universal explanation of aging. So you can study aging across epigenetics, omics, biobehavioral, or even brain clocks. And all of these are relevant. I think that we need to put the focus not in a universalistic model, but a model that can predict different outcomes with different clocks. And it's also to understand, more simple things. How, we can use aging clocks, for instance, to assess the impact of lifestyle or protective factors like multilingual limbs. In this case, we've shown that it's associated with less accelerated aging or more delayed aging. We also shown that, healthy activities like, dancing or doing music or doing visual arts or even gaming, some kind of gaming can be also accelerated with delayed brain aging. Also exercise, we have shown in a randomized control trial that the people that do, resistant exercise more frequently tend to reduce the pace of the brain aging. This is another study with multilingual and individual level analysis that we are now, it's now under review. We can use very simple metrics, like in this paper, just accepted in Science Advances, we have using natural speech analysis. We extract more than 400 features from the natural speech, and we used that to predict the age of the individuals. And we were very able to predict the age, but also not only to predict the chronological age, but to identify the individuals that have a more accelerated or delayed aging in terms of epigenetic clocks. We use Hannum, omics, and retros. There are three typical systemic, biological systemic clocks, but also brain clocks, as well as we were able to predict with these speech aging gaps, clinical and cognitive assessment, and distinguish between patients with, accelerated aging and as well as patients with dementia. Another project that I just started, I'm very excited, is that we are going to explore the relations between the exposome and the microbiome. In the microbiome, we will use the SPIRE. It's more than 70,000 subjects across 61 countries, if I am not wrong, in which we will study the neuroactive components of the microbiome. Those are pathways, genetically oriented pathways that are associated with inflammatory, immune, and other physiopathological pathways. We will estimate the clocks with that, and we will explore the association of the exposome, how, let's say, climate change, temperature, and even socioeconomics inequalities impact, increase, or reduce the neuroactive components of the microbiome and the related aging clocks. I think that this kind of, tailored, personalized approach, it's really important to understand global settings. Like most that we know come from the global north. So 90%, I will say that we know about brain health come about the 10% of the population. But, this is if we really want to move into more precision medicine, more personalized medicine, I think that we need to incorporate other kinds of information. Like, for example, the Lancet Commission have established that there are 14 factors that are associated with dementia. Those are modifiable risk factors. But, when you look at the global south, it seems that there are other factors that have a larger impact, like exposure to infections, environmental hazards, early life adversities, malnutrition, or social determinants of health seem to have a much larger effect in the global north, and they are not incorporated in the Lancet Commission. So this is an example, it's a very simple example of why, for policy reasons, we need to develop, more embedded, situated models in the context where health and disease develop. In this paper, we just included, income inequality, poverty, infections, and wealth shocks to show that the typical Lancet Commission reports show very different modifiable risk factors with a huge impact on some of them. Also, we performed Markov analysis of the impact of monoclonal antibodies in dementia in global setting, and we showed that income and exposome really increase or reduce the impact of monoclonal antibodies across countries. This Lancet piece will come up in 2 weeks, I think, in which we really propose to developing a kind of more ecological approach to risk factors, like, increasing risk architecture, like, as I mentioned before, including other exposures that could be relevant, using risk classification standards, going beyond the classical risk factors, understanding how poverty involves a kind of non-linear interaction between multiple factors, right? And for that, we need really exposome-informed modeling, like going beyond isolated additive clustering, or, the path, the typical factors that are being analyzed in the Lancet Commission through a non-linear model. So, for instance, we are starting to use a model of entropy to understand the non-linear interactions between different factors. We need to broaden also the evidence standards and to starting to expand this model into global settings and underserved populations. So understanding some underserved population, assessing people in the global north are also minorities, people in the global south and also minorities, and, people with more challenges in the environment is essential, not only for an ethical imperative, for a more precise science understanding, for being able to predict why a person with a genetic condition or vulnerability will develop, quicker or slower, accelerate aging, for example. And for that, we really need to move beyond the traditional social determinant of health and including much broader, extended set of factors, as well as much broader and extended set of measure of what is healthy aging, instead of looking just to a cognitive decline or risk of dementia, we may want to look at multi-organ clocks, brain age gap, pace of aging, and also, other advances we have been discussing today. I think that the cultural exposome also deserves attention. We are more prone to focus in environmental factors that are physical, like pollution, right, or nanoplastics, or temperature, because they seem to be more physical. But or brain systems responds with inflammation to both physical toxins and also social toxins. So understanding social factor is crucial for understanding, for example, sleep. Sleep have a huge association with exposomal factor and socioeconomic factors. And also, even societal values, um, like, well-being, social life, institutional confidence, rights and choices seems to have association with the pace of accelerated aging. So I think that we need to expand the idea of exposome. This is also something that we are going to discuss in a forthcoming Lancet Commission of Arts and Health that we are going to publish on September during the UNGA meeting, in which we really need to understand how cultural engagement, society, and arts can help to reduce the stress, and in which aspect they can really help and in which ones they cannot. So to summarize, I think that uh, if we really want to develop precision brain health, we need to develop better models of health, disease, and prediction that connects whole body with the environment. For that, aging clocks, not just aging clocks in terms of classical epigenetics, but also aging clocks of brain cognition, behavior, language, physiology, are really important because they can capture not only the cumulative burden, but also the protective and resilience aspects. We need to understand the neuroscientifics, how environments have, non-linear causalities. Sometimes they have, like kind of lifespan effects that cannot be understood in isolation. And we need to develop global consortia, as many of us are doing, but also we need local approaches because global consortia provides a level of granularity that explains, global difference, but will never tackle the precision of different factors that interacts in local settings. So we need to combine both. I think that this is the most important message that I want to share about the exposome and the SynerCom, and would love to chat with you if you have any questions, and thank you so much.
Thanks so much, Augustin. Yeah, a great presentation. I'll open it to the audience if there's any questions. I can't see any questions. So let me ask you one question, actually. So we face it. So, in like our research, we probably more focus on one of these exposomes. And the question becomes how to adjust for the other effects of the exposome. So what do you suggest? Like how, if we are going only for cardiometabolic exposure on the brain, how to adjust for the other things, like, and let's say in a general, like modeling kind of, not any like machine learning or.
Yes, yes. So thank you so much for that question. I think that there are 2 or 3 or 4 different approaches. One is the one that you exactly say, when you have a cardiometabolic effect, when you have, let's say, a pollution effects, you can do the standard approach. That means covariate for as much as possible variables that you have, right? And this is very important. I would say most of the research just don't do that. Just they do, some kind of covariate. But, pollution is usually associated, the level of exposure to pollution, it's have a kind of association with the socioeconomic level. So the people that live around fabrics are people with lower resources. So we need to control for that. This is the basic thing. And this happens across many different exposomic factors. So the people that have lower education also tend to have, a worse overall health access as well. So for that, they are the standard approaches. You can do covariate, you can do multicollinearity, you can, identify your TIF factors, those are explaining the variants that are more extreme, and you can try to dissociate the variants, right? This is a standard approach that still needs to be done, I would say. It's very, very poor in that regard. But for that, there are some challenges. One of the challenges to get access to the data. Of course, you can do exposome metric if you have zip code or some level of geolocalization. Nowadays, there are 200 different metrics, well, depending where you live, but there are many environmental and social factors that at the very least you can start to, to incorporate into the models. But the other problem with that regard is that by controlling the other effects, we are implicitly assuming that those effects are independent. And this is the challenge, right? So when you say cardiometabolic factors, all cardiometabolic factors have a huge impact, a huge association with poverty or pollution, for example. So we need to understand that some of these factors are not independent, are not individual components. They are part of multiple environmental factors that happen in a simultaneous, in a synergetic way. So for that, we need better models. Of course, you can do machine learning, but machine learning is very limited. I mean, most of the people thinks that the machine learning do fancy nonlinear things. That is not true. Most of the machine learning models are not robust to capture real nonlinear interactions. Then you have some specific models like generative additive models that they can tell you the level of nonlinearity, like level 1, level 2, or level 3. Those are the important things to understand, the mass effects. What happens when you have not one or two factors, when you have 4 or 5? Probably you have a quasi-exponential effects. Right? So these methods are robust for that. But then you have much more nonlinear and interactive models. Like, for instance, you can develop metrics of entropy, like OINFO, for example, can estimate the kind of synergetic and redundant information across multiple predictors. So you can understand kind of variance associated to having multiple factors, multiple predictors that cannot be explained by any single one. So we are starting to do this kind of analysis, and there are other groups that are starting to explore that. But I think that the main challenge is being able to assess the synergetics, the interactive effects of the predictors and the nonlinear effects. But of course, the other obvious problem or challenge is data, how to get this massive amount of data, right? So we can talk about that, but I don't want to talk too much.
Thank you. Yeah, thanks. I don't see any other questions, actually. Oh, there's a question in the chat, I think. Just a thank you.
Yeah, okay.
Okay, I think, yeah, we don't have any other questions, so we'll adjourn the meeting. Thank you really for great talk again. Really appreciate your time, so thank you, Agustin.
Thank you, Fadi. Happy to follow up, happy to explore any collaborations, and if anyone wants to follow up, just send me an email.
Of course, thank you, thanks.
Thank you, bye.